Muscle tissues. Plan. Number 1. General morphofunctional characteristic and classification of muscle tissues. Number 2. Skeletal muscle tissue. Number 3. Cardiac muscle. Number 4. Smooth muscle tissues. Muscle tissues are a group of tissues having different origin and structure, but united on functional sign contractility. They provide moving of whole organism in space and moving of different its parts and organs inside of organism, for example, heart, tongue, intestine. For all groups of muscle tissues are typical the following signs: elongated shape of their structural components, presence of longitudinally located myofibrils, special organelles providing contractility, location of mitochondria near contractile elements, presence of inclusions of glycogen, lipids, and myoglobin. Classification of muscle tissues. There are two classifications of muscle tissues. One is morphofunctional, the second is histogenetic. Morphofunctional classification. Cells of muscle are called striated or smooth muscle depending on the respective presence or absence of a regularly repeated arrangement of myofibrillar contractile proteins, the myofilaments. Striated muscle cells display characteristic alternations of light and dark crossbands which are absent in smooth muscle. Histogenetic classification is based on sources of development. According to this classification, there are 5 types: mesenchymal type, derived from mesenchyme; epidermal, derived from skin ectoderm; neural, which source is nerve tube; coelomic, derived from ventral mesoderm; and somatic type, which source is dorsal mesoderm. Striated muscle. There are 2 types of striated muscle: skeletal muscle, accounting for most of the voluntary muscle mass of the body, and cardiac muscle, limited almost exclusively to the heart. Skeletal muscle tissue. Skeletal muscle tissue is derived from myotomes of somites. Skeletal muscle tissue composes approximately 40% of the total body weight. Skeletal muscle tissue is basic tissue of skeletal muscles. It has symplastic type of structure. The structural unit of skeletal muscle is muscle fiber. Muscle fibers are arranged parallel to one another with their intervening intercellular spaces, housing parallel arrays of continuous capillaries. Each skeletal muscle fiber is long, cylindrical, multinucleated, and striated. The diameters of the fibers vary, ranging from 10 to 100 micrometers. From the outside, muscle fiber is covered by sarcolemma, including basement membrane and plasmalemma. Muscle fiber consists of 2 components: myosymplast and myosatellites. Myosatellites. Myosatellites are slightly differentiated one-nuclear cells. They are located between basement membrane and plasmalemma of muscle fiber. They contain common organelles, do not contain special organelles. Myosatellites are source of regeneration of muscle tissue, of skeletal muscle tissue. Myosymplast. It has number of oblong nuclei located at the periphery, immediately under the sarcolemma. Number of nuclei in myosymplast can reach a few dozen thousands. The basic part of myosymplast, myofibrils, compose. Myofibrils. Myofibrils, the contractile organelles of skeletal muscle consisting of thick myofilaments with protein myosin and thin myofilaments with protein actin. Myofibrils are about 2 micrometers in diameter and extend the entire length of a muscle fiber. Their prominent striations make the entire skeletal muscle fiber appear striated. A fluid-filled system of membranous sacs called the sarcoplasmic reticulum encircles each myofibril. dilated end sacs of the sarcoplasmic reticulum called terminal cisterns, but against the T-tubule from both sides. T-tubules. In some parts, plasmalemma of muscle fiber forms protrusions like tubules which pass perpendicular to muscle fiber via its thickness. These are T-tubules. A transverse tubule and the two terminal cisterns on either side of it form a triad. Triad of skeletal muscle fiber. So longitudinal cisterns of smooth endoplasmic reticulum from both sides pass through T-tubules, fit closely and form terminal cisterns. In a relaxed muscle fiber, the sarcoplasmic reticulum stores calcium ions. Release of calcium ions from the terminal cisterns of the sarcoplasmic reticulum triggers muscle contraction. Within myofibrils are smaller structures called filaments. Thin filaments are 8 nanometers in diameter and 1 to 2 micrometers long, while thick filaments are 16 nanometers in diameter and 1 to 2 micrometers long. Both thin and thick filaments are directly involved in the contractile process. Parcel of myofibril containing just thin actin myofilaments is called eye disc, light isotropic. Via the middle of eye disc, Z-line pass. It is called telophragm. During contraction, eye disc is decreased or disappears. Parcel of myofibril containing thick myosin filaments form A disc, dark and isotropic. Via the middle of eye disc, M-line pass. It is called mesophragm. Sarcomere. The parcel between two telophragms is called the sarcomere. Sarcomere is structural functional unit of myofibril. Formula of sarcomere is 1 second of eye disc plus A disc plus 1 second of eye disc. Connective tissue investments. The connective tissue investments of skeletal muscle are the epimysium, perimysium, and endomysium. The entire muscle is surrounded by epimysium, a dense irregular collagenous connective tissue. Perimysium, a less dense connective tissue, surrounds bundles or fascicles of muscle fibers. Endomysium, composed of reticular fibers and an external lamina, surrounds each muscle fiber. Because these connective tissue elements are interconnected, contractile forces exerted by individual muscle cells are transferred to them. Tendons and aponeuroses, which connect muscle to bone and to other tissues, are continuous with the connective tissue encasements of muscle and therefore act in harnessing the contractile forces for motion. Cardiac muscle tissue. It is derived from visceral layer of mesoderm, splanchnic mesenchyme, the myoepicardial mantle. It is a coelomic type of tissue. Its structural functional unit is cardiac muscle cell or a cardiomyocyte. Cardiomyocytes are connected with each other by intercalated discs and form functional muscle fibers. Elongated cardiomyocyte contains single centrally placed nucleus and longitudinal striated myofibrils at the periphery. Striated myofibrils has same structure as in skeletal muscle tissue. Here in the slide you see enumerated by 3 intercalated discs. Cardiac muscle cells form highly specialized end-to-end junctions referred to as intercalated discs. Types of cardiomyocytes. There are 2 types of cardiomyocytes, contractile and conducting. In this slide you see contractile cardiomyocytes enumerated by 1. During long work For example, in conditions of constant arterial pressure, working hypertrophy of cardiac muscle cells occurs. Stem cells or cells predecessors of cardiac muscle cells are absent in muscle tissue, so dead cardiac muscle cells are not regenerated. Smooth muscle tissues. There are three groups of smooth muscle tissues: mesenchymal, epidermal, and neural. Smooth muscle tissue of mesenchymal origin. Localization: wall of internal organs, wall of blood and lymphatic vessels. Its structural functional unit is spindle-shaped smooth myocyte. Cytoplasm of myocyte contains slightly developed common organelles placed in poles of stick-shaped nucleus located at the center. Smooth muscle tissue of epidermal origin. The source of development is skin ectoderm. Localization: terminal sections of sweat, mammary, salivary, lacrimal glands. The structural unit is star-shaped myoepithelial site. Contractile apparatus is located in processes of the cells. Contraction of processes leads to excretion of secretion from the gland. Smooth muscle tissue of neural origin. The source of development is neuroectoderm. Localization: two muscles of eye iris, dilator pupil and sphincter pupil. Structural element is myoneurocyte. Thank you for attention.